So this is going to be a short video which would help you to go through some of the practicals before your alternative practical paper. This is directed towards the 2026 May-June paper that is coming up in a few days' time. So if we think about your paper 2, there were only a few sections that was being tested at this point. So based on that, we can also assume what may come for the exam. And you need to understand that this is not a prediction whatsoever, but this is just an analysis of some of the practical papers that have been there in 2025 and in 2024. And also we will be looking into some of the practicals that have not been tested for some time. I'll be using your syllabus. This is the latest edition of the syllabus, by the way. In this syllabus, if you go to page number 9, it basically talks to you about what your practical paper is like. So you know it's a 1-hour paper and you are to get 40 marks for this particular paper. Now, unlike the other papers, this is one of the most easiest papers that is there, because most of the practicals are the same. If you can understand what kind of theory that they do test you, it makes it very easy to score at the practical paper. As you can see in this syllabus, they say that the questions will be based on the experimental skills in section 4. So in your syllabus in section 4, they do speak about a list of practicals that is given over here. So you can see this is page number 50 in the PDF. They give you the list of diffusion, osmosis, and you could see they even talk about the different types of food testing. And this is the whole bunch of list of practicals that you should have done in your coursework, or at least you have done the theory behind how this practical is done. So what we will do in this lesson, as I said before, we'll talk about certain areas that have been tested in the recent papers, and based on that, what kind of questions can you really expect at your own paper. So in your syllabus, we start off with the characteristics of living organisms. Now, one of the most recent papers had a picture of a starfish, and they basically ask you to do a biological drawing of it. And then you had to calculate the magnification from that. Now this is very different to what we saw in the previous papers, where most of the questions were about a leaf and you need to draw a cross section of a leaf. So this means that you might want to be ready with another type of biological drawing, because in the older papers, this is like in 2010, 12, those areas, you get to see a lot of variation when it comes to the biological drawings rather than the standard leaf that you have been seeing in the recent papers. So I would say you might want to look at some of the pictures of some of the kingdoms that is mentioned in this first lesson, and know a little bit about their characteristics, just in case you may be asked a question about how did you identify the particular kingdom of this organism in the practical paper. Because these are also practical knowledge you should be having. So my suggestion to you would be try out a few questions, try out a few biological drawings of animals, plants, or any other variations that they had given, not only the leaf structures that you have seen in the recent papers. So my advice to you would be to go through the structures of these a little bit and know a little bit about the functions of those structures too, in case they do ask a 1 or 2 mark question on that part. So that's what I feel that you can get from the first lesson. And as you know, there's always a question when it comes to magnification as well as in biological drawings. Now you have to be very careful at your paper, because they may just ask you to draw a large drawing of whatever the specimen that is given to you. Sometimes the question might ask you to draw maybe just a single cell from the entire specimen, or just a small section that would be given in a box or something. You need to read the instructions very carefully and draw according to the instructions. Now in some of the biological drawings, they expect you to label the diagram as well, and in most cases they would say draw a large labeled drawing, which means labeling will take some marks for you as well. So do pay attention to all that. And as you know, whenever you do a biological drawing, you need to have a pencil, which is HB, that is the standard. You need to carry a sharpener with you, as well as a good eraser. Make sure your eraser is clean, because you don't want any stains on that paper when you're doing this. You also need to have a ruler, because when you're labeling the diagram, you need to use rule lines. Remember, you do not use any arrowheads at this point, and your line should be drawn horizontally, not everywhere around the diagram. Then coming back to the cell structure, in your syllabus they basically talk about you needing to have drawings of certain parts within the cell, like the mitochondria, the chloroplast. You should have been seeing photomicrographic pictures, that means real pictures of these structures under the microscope. So you could be given any type of biological drawing like that, and you might have to draw an enlarged diagram of a part within the cell, which means you need to still brush up a little bit about the functions of these parts as well. So these are possible areas. I've not highlighted this in this part, but let me highlight it over here. You need to know these organelles in case they give you a picture under the microscope. You should be able to identify them, and you should be able to draw them as well. Another area that was not tested for a long time in terms of biological drawings are the specialized cells, because there had been a lot of past papers about 8 or 10 years ago from these specialized cells where the children had to do biological drawings. So you might want to look at those cells maybe on the internet and be familiar with what those pictures would look like. Talking about this magnification, most of the recent questions had been about you're given a specimen, you draw it, then you'll get to measure the specimen and then calculate the magnification. This is very similar to what I'll be showing on the screen over here. You can see this from a past paper where a particular insect or a beetle was given over here, and the children were asked to draw a large drawing of this entire beetle. And just like what you see in the recent papers, you're supposed to calculate the length, not calculate, rather to measure the length of your drawing as well as in the picture, and then using the formula for magnification, you just had to calculate it. But there are also other types of magnification type questions that has been in the past, and one of the types of this is what you see here in the picture, where you could be given a picture like this, and you may have noticed they have even shown a magnification underneath the diagram that is given to you. Now what does this tell you? The specimen, this blood smear that you're looking at this moment, has already been magnified by 800 times. So whatever you draw is a magnification of another magnified image, right? Magnified specimen, that is. So when you're calculating the magnification, you need to understand, you need to consider this magnification of this specimen as well. Now the usual way we calculate the magnification is the drawn image divided by the actual image size, and that's your magnification. But if you are to calculate the magnification, let's say of this cell C, the red blood cell in this picture, well, you need to first do the same, drawing, measure your drawing, measure the size of the red blood cell shown in this picture, but you need to multiply that by 800, which is the magnification mentioned at the bottom of this picture. Now this is something that has not been tested very recently, so that's something you might want to look out for. Another type of magnification question, which again has not been tested for a while, so this is actually a pollen grain that you're looking at, and they have given you a scale bar at the bottom of this picture. and you can see they have mentioned a certain value which is 0.05 millimeters. Now in a question like this, as you can see, they've not even asked you for a biological drawing. They straight away ask you to calculate the magnification. Now this is where lots of children don't understand the purpose of the scale bar. So in case if you do get a question like this, you need to first measure the length of the scale bar using a ruler. Measure from one bar end to the other bar end, and let's just assume that the length of your bar came to 2 centimeters. So what does this tell you then? 2 centimeters anywhere in this picture is in real life 0.05 millimeters. So now you consider your drawn image to be 2 centimeters, because that's what the bar says, divided by the number that is mentioned here at the bottom, because that is considered as the actual size of the specimen. So this is something that has not been tested for a while. So you might want to go through some questions like that. There's only a few of them in the past papers also. So just in case, please go through this section as well. Okay, so going back to the syllabus, you also might know that whenever you are to take any measurements in the practical paper, your answer should be given in millimeters. This is a standard and it has been mentioned in the syllabus. You can see I've already highlighted this for you. But if and so they do tell you, look, we need the answer in micrometers, then you should be able to convert that into micrometers or another unit that they're asking you to keep the answer in. But if not, the standard is to leave it in millimeters. You might want to check when you're writing your answer, have they already given you the unit? If not, you need to mention the unit as well. So in terms of the lesson on movement in and out of the cell, if you take the recent papers, there had been about a potato strip being put into a different solution and they're measuring the change in length, questions based on that in the recent papers. Now, one of the things that they have not tested for a while is about factors that affect the diffusion, about surface area to volume ratio. How does temperature basically affect the rate of diffusion? Now, one of the classical practicals we do or we teach the students is to take an agar block, usually with a chemical like phenolphthalein inside these agar block. And what do? We drop this into an acid like hydrochloric acid and we measure the time it takes for the block to become colorless once more. So this is a classic experiment when it comes to diffusion, distance and all that. This is also something that you can see in your theory, in your notes that you may have received, or even in your textbook as well, where you probably might be asked to calculate the area of the cube, the volume of the cube, and then calculate the surface area to volume ratio. So you might want to go through the theory behind this to understand how to do a calculation like that. Also in this particular section, under osmosis, there are lots of questions that can come under this. And one of the things that they have said over here is investigating osmosis using materials such as dialysis tubing. Now, dialysing tubing is similar to what we call as whisking tubes, which is a partially permeable membrane. So there are lots of questions based on this once more, where maybe a sugar solution is put into the whisking tube and it's basically tied up from the end, it looks like a sweet basically when it's tied up, and it can be put into distilled water or maybe another concentrated water. So what happens is you measure the mass of the whisking tube with your solution at the beginning, and after the experiment they could ask you, what is the final mass that you get, or they might give you the data and ask you to draw a graph based on it, right? And they could also ask you to find out what is the water potential maybe in the solution inside the whisking tube. Now this is again a very classic experiment where you probably might be given the data to plot on a graph, let's say for example the change in mass of the whisking tube with the concentration of the solution that you are changing. Now if you do draw a graph like that, you will see that your line would basically pass through the x-axis. Now the point at which the line passes through the x-axis means that there is no change in the mass of your whisking tube. This basically means that the water that came in is equal to the water that went out, or in another word, there is no net movement of water. That means that is equivalent to the water potential outside. So this is an experiment that you could get where you're given the data, you plot the graph, and then you estimate what the water potential might be. Another type of questioning you can get on osmosis over here is where they could put a sample of maybe red blood cells or some sort of animal cell in different solutions and give you an actual picture under the microscope and ask you what has happened in this, like has water gone into the cell or has water gone out of the cell. If it's in the case of a plant cell, then you might want to know words like turgidity, flaccid, plasmolized. I know it's coming under the supplementary section, but you still might want to go through those words and be familiar what those cells would look like under different solutions. I mean, it's very unlikely because these kind of questions usually come in the theory paper, but looking at the structures, how it has been changing with time now, you might still want to brush up on a little bit of these theories as well. Then going to biological molecules. So this is an area which has been tested very little in the recent papers. So you might want to brush up on the different types of food testing, the safety precautions, and you might also want to go through what are the reagents that you're using. Now in the previous year and the year before, already they have talked about iodine test in one of the potato experiments with osmosis as well. They've also talked about the Benedict's test and a little bit about the ethanol test as well. Now this does not mean that they will not come, but what have not been tested very recently is the biuret test as well as the DC-PIP test for vitamin C. Now one of the ways a question could come from vitamin C is for you to design an experiment. Let's say for example you're given maybe an imported mandarin and a locally grown, let's say a Sri Lankan mandarin or so, and you want to estimate or you want to find out which of these has the highest concentration of vitamin C. So you have to design an experiment now using DC-PIP to see what may have more vitamin C than the other. So obviously this is going to be a qualitative test where you observe and you make your decisions, but you need to know that DC-PIP is a blue color solution, and due to vitamin C it basically turns colorless, which means the higher the vitamin C concentration, the faster the color change will happen. So you should be able to design an appropriate experiment just for that purpose. Going into enzymes, well, enzymes is an area again that can always come in the practical paper. Again, something to do with designing of an experiment where you might want to be able to design to show how temperature affects the rate of a reaction or how pH affects the rate of a reaction. Now when it comes to temperature, you need to always keep in mind how do you maintain the temperature throughout that experiment. You need to use a water bath, and if it's a thermostatically controlled water bath, it's much accurate because you know it's going to keep on maintaining that with less fluctuations taking place. If it's something to do with pH, you need to mention about using buffer solutions when you're designing these experiments as well. Now, talking about designing of experiments, you also need to remember that when you're writing the method of the experiment, you need to have at least 5 different variables. That means, let's say for example, if you're changing the temperature, you need to have a minimum of 5 readings in a set. And to increase the reliability, you want to do the experiment at least 3 times to increase the reliability as well. So you need to talk about all of these when you design the experiment. Going a little bit further about plant nutrition, so this is about photosynthesis. Now, in terms of plant photosynthesis, again, there had been many questions in this section where you have been given a leaf again, or you have been tested about using an indicator like hydrogen carbonate. Now, this was there in the 2025 papers, using a water plant and hydrogen carbonate indicator to measure the rate of photosynthesis. Now, you might still want to go through the theory of this, as I said before. Another thing is to find out the effect of light intensity, CO2 concentration on photosynthesis. Again, you can use a water plant for this to measure the rate of photosynthesis. And most of the time what we do is we measure the number of bubbles that are evolving during this reaction. But in your theory class, you may have learned that without counting the bubbles, we can also measure the final volume of oxygen collected, which is much more accurate because counting bubbles have lots of problems when you do it. So you might want to list down some of those problems you face when you do an experiment like that. Also going a little bit further in this, as I said, that lots of biological drawings have been aimed towards the leaf and the photosynthetic part of that. But one of the things that I would say I'd like to see in a picture nowadays would be to have a photomicrograph, a real picture of a cross-section of a leaf, and get the students to identify them, and maybe ask the students to magnify one of the cells in that, and then ask the function of it, calculate the magnification of it. So those are some possible areas that you can expect from this particular section. Another area where you can be tested here would be a cross-section of a stem or a root, and ask you to identify what those cells are in the cross-section. Now, most of the time when we learn about transport in plants, we do a hand drawing of the cortex and everything, but most of us, we do not go through the photomicrographs of a cross-section of a stem. Now, as you can see in your syllabus, they've already mentioned about these different parts as well, here in the photosynthesis lesson as well as in the transport in plants. So you might want to go through the cross-section of a stem and a root and identify all the parts mentioned in your syllabus. So in terms of another area that you can be tested over here is to be given you a picture of an actual tooth, maybe a human tooth, and ask you to identify what that tooth is, maybe ask you the function of the tooth, and they could ask you to do a biological drawing of a tooth as well. Now, if I'm not mistaken on this, I remember in 1994 or sometime a bit later on, there was some questions regarding these human teeth as well. So you might want to check on the internet if you can locate what these questions are, and try to practice drawing these teeth, because this is another section that has not been tested for a very long time. Including a cross-section of a tooth as well. So if they can give you a good cross-section of a tooth with good lighting, you might be able to see the 3 layers of the teeth as well. Another possible area that you might want to check out, as I said before, is the transport in plants of both a root and a stem. You should be able to label those parts, because as you can see in this theory part about the uptake of water, they've also mentioned about the different sections within a cross-section of a root as well. In terms of transpiration, I know that there was a question in your paper 2 this time about the potometer. So it's very unlikely that you would be tested again on the potometer. But then again, there have been some practicals, which is mentioned even in your textbook, about using 4 different leaves with Vaseline applied both on the upper surface and the lower surface, and then have one on the upper surface and on the other one on the lower surface. Then you basically measure the mass of the leaf, and then thereafter you can measure the final mass of the leaf after transpiration. that is to find out where most of the stomata is found in a leaf. So that kind of a question can still come for you, which checks your theory knowledge and your designing of a practical as well. In terms of transport in animals, one of the things that I think that you already got a question about the circulatory system, but nevertheless, you still can be given a question about a cross-section of a real heart and probably ask you to identify the parts of it, or maybe draw a small section of a heart. Now, that kind of a question again came in the year 2000, that means 2002, 2003, like, where a heart was given and a box was drawn in a section of the heart, where the children had to draw a part of a ventricle with its valves and all that, which was one of the toughest biological drawings that I have seen before. So that kind of a question is also possible again. So you might want to look through a cross section of a heart, a real heart by the way, which you might get to draw. Then a lot of questions in the recent papers have talked about investigating and describing the effect of physical activity on the heart rate. Basically, you have to design an experiment to prove this, where you think about a particular exercise, get the people in your group to do the exercise, repeat the experiment, take the heart rate values and all that. Now, this has been a bit of a favorite question in the Cambridge past papers as well as in the Pearson past papers as well. So you might want to just go through it again with some marking schemes. It's very unlikely this time to get a question like this, but you still might want to run through it. Also, another area which I feel that has not come for a long time is a real picture of the different types of blood vessels, asking you to identify which blood vessels are shown in a particular diagram, how did you identify them, and they could even ask you to do a biological drawing once more in this section as well. So this is again a section that you might want to go through and train your eyes to recognize what these blood vessels are. Then in terms of blood, according to the syllabus, it says that identify red and white blood cells in photomicrographs. According to the supplementary syllabus, you should be able to identify photomicrographs of both lymphocytes and phagocytes. So even if you are doing the core syllabus, I would still advise you to look at a blood smear and try to see whether you can identify the different types of blood cells. Maybe draw a small diagram of it and see whether you know how to draw it as well, right? So these are sections to look into. In terms of disease and immunity, I don't think that you can actually get a question on this section. However, in gas exchange, you could be given a real picture or a cross section of a lung, where you might have to identify the alveoli. They could ask you to draw a small section of it, and they could also ask you how it's adapted for gas exchange in terms of a theory question, right? Again, this number 5 about physical activity and the rate of breathing again has been a bit of a favorite question in the practical papers as well as in the Excel papers as well. I feel it's very unlikely to get a question this time on it, but as I said before, you might still want to go through some of the past papers as well. Again, in terms of respiration, there had been a lot of questions on respirations also in the recent years. But one of the things that they have not tested is to investigate how temperature affects the respiration rate in yeast, where you basically put yeast into a sugar solution and you basically measure the height of the foam that is forming on top. So that's one kind of a practical that you still can get. And again, because this is temperature, you might want to use some water baths and mention the 5 different temperatures that you're going to realistically use in this experiment. In terms of the rest of the syllabus, coming to coordination, this is one of those things that you could get in terms of a question in coordination would be to identify a sensory neuron, a relay neuron, and a motor neuron. Relay neuron again is very unlikely, but a sensory neuron and a motor neuron you should be able to identify if you're given a picture, and label the parts as well. Now, in terms of coordination in the past papers, there had been questions like a cross-section of an actual eye, and children have to identify it and draw a certain section of the human eye as well. Now, that is something that we have not seen that for a very long time in past papers. So something that again you might want to brush up in terms of theory again. Again, in terms of coordination, they could ask you to design a practical. I remember in some of the past papers there was like a compass like thing where you have two needles at end, and you need to keep it on your palm and measure when do you last feel the pain. Like you basically keep reducing the length between the needles and you measure at what point you feel the pain again. So they have given a practical like that, and they have asked you to design how do you measure this, what are the problems that you face. So you could be asked to design something that you have never learned before. So you need to use your practical skills mentioned in this syllabus. Please go through that, by the way, and you should be able to design a practical. Then in terms of the other sections, again coming to plants, the tropic responses, they could ask you about this experiment about phototropism and gravitropism. This usually comes in your practical paper, but again they could ask you to design a practical on how you're going to find out the effect of maybe light intensity or something on the phototropism, something like that. Yeah, so that's about it, I guess, in the practical paper that you really can expect to get in terms of questions. As I said, you need to keep on practicing your biological drawings, and I think you also might want to try some papers before the year of 2015, because there were lots of various types of questions, because you can see in the 2025 papers the questions have been very similar to each other, right? And you also need to know how to draw a table properly, because one of the common mistakes children do when they draw a table is they forget to have the independent variable on their left hand side and the dependent variable on the right side. Another common mistake children do is they do not put the units at the headings of the table, and they start putting those units in the place where you put your measurements or your observations. When you are drawing the graph, the standard way of marking a graph is usually a 4 or a 5 marker, where we will look to see whether you have marked the x-axis and the y-axis along with its units. Have you marked the points on the graph properly using either a cross sign or a dot with a circle around it? Have you done any extrapolations in your lines? That means when you're joining the points, have you extended the graph when they have never asked you to do that? So you have to be careful with those sections as well. And also you need to go through the different types of graphs. Why I'm saying this is because most children do not know the difference between a bar chart and a histogram. Because in a bar chart, your bars have to have the same thickness and the same distance between them as well. If it's a histogram, you need to have bars of the equal thickness, but they should be together. And very recently, again, if I'm not mistaken, this was in the 2025, but it was, I think, February, March or May, June. There was a question where you had to draw a histogram. And in most cases, children went and read this as a normal bar chart, which means they do not know the difference between them. So going back to the syllabus, if you just punch in number 50, here they have talked about all the practicals that you are to know the theory or to do this in school if it's possible. They've also talked about what are the knowledge that you should have in the practicals. So I hope you will go through these sections as well, right? So I guess that's about it. You really need to be worried about the practical paper. So please take all the requirement things that you required for the practical paper, including your calculator as well. Please make sure that you stick to the time. You don't get caught in your biological drawing for more than the time that you should be spending on it. Alright, so I hope this video helps you to go through at the last moment what you need to go through from your practical paper. So if you like this video, please put a thumbs up, and if you would like to subscribe, please do subscribe to my channel as well. Thank you.